Okay, so this topic is about hematopoiesis still, but this time around, it would be the lineage-specific hematopoiesis. So what you've had from your previous discussion was the general characteristics of hematopoiesis. So you've had the stages in hematopoiesis, right? So you have your mesoblastic, your hepatic, and then your medullary stages. So what's important to remember in hematopoiesis is if you say hematopoiesis, it should be a controlled and regulated process of producing cells. Remember, your body will produce what you need. So that's why if you want to produce these cells, you will need certain stimulants or growth factors that can help you initiate the production of blood cells. Okay? So today, it will then be about the specific production of your blood cells. So we'll have the different maturation stages for RBCs, for your WBCs, and a glimpse of platelet production. You'll further highlight that one when you go into your HEMA2 class. Okay? And just a short recall, remember, in hematopoiesis, as to theories, it all starts from a pluripotential hematopoietic stem cell. Okay? So as to theories, all these blood cells will then be derived from that pluripotential hematopoietic stem cell. And as you can see here in this picture, as to theories, later on, it will then give rise to the formation of your progenitor cells. So you have your lymphoid and your myeloid progenitor cells. And this unrecognizable hematopoietic progenitor cells on the other end can be divided into two major types. Yun nga, yung committed progenitor cells natin, and the other one is the non-committed progenitor cells or non-committed hematopoietic stem cells. So you have their committed myeloid progenitors. Yan, we collectively call it as CFU-GEM. Okay? Committed because later on, they can then give rise to multiple lineages of your blood cells. Okay? So later on, as we go along, we'll then deal with these specific stages in hematopoiesis. Okay? So just to give you an eye view, so your hematopoietic stem cell here, so you have here your pluripotential hematopoietic stem cell. So as to studies, ayan, we have quite a limited number of hematopoietic stem cell in our bone marrow. But the good thing is, these hematopoietic stem cells, remember, are self-renewing. Okay? And we can come up with billions of blood cells from these hematopoietic stem cells within our entire lifespan, generally. Okay? So from this pluripotential hematopoietic stem cell, remember, there are certain stimulants or growth factors that can stimulate or help you produce out the blood cells. Yan, we term it as positive influence. So when you say positive influence, meaning to say, you can produce cells. Okay? It will help you produce your cells. So you have here your growth factors or your stimulants or positive influencers like interleukin-1, yan, interleukin-3, interleukin-6, interleukin-9, sino pa ba? Interleukin-11. You'll also have their KIT ligand, and you also have there your GM-CSF or colony stimulating factor. It's very important to remember that you have these stimulants, you have these positive influencers because they will help you come up or produce your mature specific blood cells. Okay? And then from this pluripotential hematopoietic stem cell, you'll have your progenitor cells. And then later on, ayan, as to studies, you will then form CFUs or colony-forming units. You call them as such because they are capable of forming colonies on semi-solid media. Ayan, sa mga experiments, sa mga culture experiments, they are capable of forming colonies on your semi-solid media. That's why you further call them as colony-forming units. Okay? Then, with your positive influencers, dagdag natin, your KIT ligand there and FLT3L can work with one another together with your other influencers like interleukin 3 and CSF and other cytokines, so you can promote hematopoietic stem cell proliferation and differentiation. So in short, basta sinabing positive influence or stimulants or growth factors, it can then help you produce out your blood cells. Okay? And the first thing that we will have here is erythropoiesis. So when you say erythropoiesis, it's a process by which your red cell precursors or red cell immature forms differentiate, they mature, so they can become RBCs. Okay? So in simpler description, erythropoiesis is the production of your RBCs. Okay? So from your stem cells to your progenitor cells to your CFUs to your BFUs or burst forming units, later on in erythropoiesis, at the end of the day, you will then produce your RBCs. Okay? And if you want to produce these RBCs, you will need stimulants. You will need your growth factor. And the main regulator of RBC production is what we call as EPO or erythropoietin. Okay? EPO or erythropoietin. It's basically a hormone released by your kidneys whenever there is hypoxia. When you say hypoxia, it's when your oxygen level in tissues is low. So it's like, it's your stimulant. So when your body senses that there is hypoxia, it will then trigger the release of erythropoietin from your kidneys. Okay? Primarily from your kidneys. A little portion could be produced by your liver, but the main producer of your erythropoietin will always be your kidneys. And particularly in your renal peritubular interstitial cells. So generally, it's within your kidneys. So ang ginagawa ng EPO natin is it binds to certain receptors by your progenitors. For example, this is my progenitor. This is my RBC precursor. When you say precursor, this is my immature forms. Okay? So let's say you have here your precursor, you have here your progenitor. So what will happen is your EPO binds on these progenitors. Okay? And then later on, as it binds to your progenitors, it will then stimulate them to come up or produce your RBCs. So it's like it will recruit more colony-forming units and then yung mga burst-forming units natin. And later on, prevents apoptosis of erythroid progenitors. So para hindi sila masira. Okay? And most importantly, through this EPO, it can induce hemoglobin synthesis. Always remember this. Red cells are not capable of synthesizing hemoglobin. Okay? What makes red cells functional, what allows red cells to carry and transport that oxygen is basically because of hemoglobin. Okay? So in erythropoiesis, if you will go on to the most important detail there, in erythropoiesis, the most significant thing that you need to do is, as you mature, as you grow, as you develop, you need to produce out your hemoglobin. You need to synthesize hemoglobin. Because without this hemoglobin, red cells are nothing. Okay? Without that hemoglobin, your red cells will not be functional. So if you want that cell, if you want that red cell to fully transport, bind, and carry oxygen, you need to have that hemoglobin. So through the help of your EPO, you can then induce the synthesis of hemoglobin. That's why later, as we go along, we should then take note of the different stages in erythropoiesis that are capable of producing out hemoglobin. Okay? And before we get into those stages, and you look at this next slide here, or before we get into those stages, in erythropoiesis, as the cell mature, there will be certain changes. O, parang ano lang yan, isipin natin, parang tao. Okay? Mula nung baby tayo, as we were born, then you become a toddler, you become uh, a student, di ba? Naging ano ka na, naging ano na kayo, school children na kayo. Then you become a teenager, di ba? You undergo adolescent changes, and then you become an adult, and so on and so forth. So parang tao lang din yan. So as we mature, as we develop, as we grow, definitely there will be changes. Okay? So the following here are the different changes as your cells mature. First things first, if you will look at the size, this would be the early stages. Okay? So as the cell mature in erythropoiesis, the overall diameter of the cell becomes smaller. Okay? The overall diameter decreases. So in short, as the cell mature, the size of that cell becomes smaller. Okay? And in relation to that, as the cell mature, the NC ratio of your maturing cells will also then decrease. Okay? Paliit na sila rin ng paliit or pababa ng pababa. And then later on, the nuclear chromatin becomes more and more coarse, it becomes more clumped, and it becomes more condensed. And as to studies, the nucleus is said to be pycnotic as later on they mature. And as the nucleus becomes pycnotic, the nucleoli inside later on disappears. Okay? And as that nucleoli disappears, it then precedes the cessation or the ultimate cessation of protein synthesis. Meaning to say, mahihinto na or ma-stop na yung protein synthesis natin. And then lastly, as the cell mature, ayan, look at the color here. As the cell mature, the cytoplasmic color changes from blue here, it turns into grayish, it turns into somehow pinkish, indicating that you have already synthesized hemoglobin. Okay? So, the eosinophilia, yung pagka-red or pagka-pinkish ng RBCs natin, remember, correlates with the accumulation of hemoglobin as they mature and as they develop. So, it's very important na in erythropoiesis, aside from these things that we have mentioned, as the cell mature, you need to create your hemoglobin. Because again, red cells are not capable of synthesizing hemoglobin. They have hemoglobin inside, yes, they carry hemoglobin inside, but the ones that synthesize that hemoglobin are your precursor cells, are your immature forms. Or later on, you'll find out why. Okay? So you look at this table here. So these are the different terminologies that you can use in identifying the different stages in erythropoiesis. So this one here usually is used in the United States. Ayan, this one here is used usually in Europe. Ayan, according to according to Rodax. Okay? So we don't have any choice but be familiar with these terms. Okay? In your exams, you may then encounter synonyms of these terms. Okay? So please go over with these terms. Okay? So to have an eye view in this erythrocyte production or RBC production, again, dagdag muna natin, it all starts from your pluripotent hematopoietic stem cell. So from this pluripotent hematopoietic stem cell, through your stromal cells and cytokines, it then leads to your, yan, sabi natin kanina, CFU gem. Okay? You will have your CFU gem. Okay? Then from this CFU gem, it gives rise to your earliest identifiable colony of RBCs called as your BFUE. So from CFU gem, it will then give rise to your burst-forming unit erythroid. O ano sir, ano yung gem na yan by the way? So that stands for granulocyte, erythrocyte, monocyte, and then your megakaryocyte. Okay? So from your pluripotent hematopoietic stem cells, ulitin natin, through the stromal cells that you have within your bone marrow and your cytokines, your stimulants or your growth factors, it then leads to your CFU gem, later on, giving rise to your identifiable colonies of RBCs known as burst-forming units. And in this burst-forming units erythroid, usually, in your semi-solid media, the colonies are large. Okay? Malalaki. Whereas in CFUs, yan, sa colony-forming unit natin, medyo mas maliliit yung mga colonies natin. And remember, your BFUEs contain very few receptors for EPO. O let's say, this is your BFU. Diba sabi natin kanina, EPO, sir, binds to my progenitors. Right? So with your BFU, yes, you have receptors for erythropoietin, but it's very minimal. You only have few receptors for erythropoietin. But under the influence of other stimulants, like your interleukin-3, CSFs, your colony-stimulating factor, your KIT ligands, later on, this burst-forming unit will then give rise to your colony-forming unit. It's like it develops later on to your colony-forming units. And this time around, as compared to your BFUs, your CFUs now will have multiple receptors with EPO. Okay? Mas marami na ngayong EPO receptors. Meaning to say, the more EPO receptors you have, the more EPO can bind on this progenitor. And as mentioned a while ago, once EPO binds onto your progenitors, it then stimulates the maturation, proliferation, and differentiation of cells. And more so, once EPO binds on your progenitor, it then helps you induce the synthesis of hemoglobin. Okay? So as to the numbers, from your BFU until it becomes CFU, as to the numbers, it may then take you roughly about 1 week for your BFU to mature as CFU. Okay? And from your CFU, yeah, look at this one, from your CFU until it becomes pronormblast, later on, it will take another week. Okay? So, another 7 days pa uli. And don't forget, when you reach this pronormoblast stage, that is then considered as the earliest recognizable stage under the microscope. Earliest recognizable stage in erythropoiesis under light microscope. So if you are examining a bone marrow specimen and nakakita ka niyan, o ibig sabihin, para mas maintindihan natin, If you see the cell, if you see the characteristics of that cell, it's like, sir, I can now predict the future. What do you mean? It means to say na, sir, upon analyzing the characteristics of these cells, I know that in the future, sir, as it further mature, I know, sir, na it will become a red cell. Okay? Kaya in cell production, yan, tandaan, you always start with the recognizable stage. Yan, kaya tingnan nyo yung mga tingnan nyo yung mga terms natin kanina. Well, you start with this. Okay? Because that's the earliest recognizable stage. So approximately, it will take you roughly 18 or up to 21 days pa to produce a red cell from your BFUs. Ulitin natin. From BFU to become CFU, it will take you about 1 week. Okay? From CFU to become pronormoblast, it will take you roughly another week. So, sir, naka 14 days na tayo. That's 2 weeks na, sir. And from pronormoblast until it becomes a mature RBC, it will take you roughly another 4 to 7 days. So if you will add up the numbers from your BFU until you produce the mature RBC, it will roughly take you about 21 days or 18 to 21 days. Okay? And as you look at this figure here, look at this one. As your cell mature, yan, starting from your CFUs, giving rise to your pronormoblast, as they mature, they undergo division. Right? They undergo mitosis. Parami sila ng parami. So imagine, from a single pronormoblast here, you start with this. From a single pronormoblast, roughly around 8 to 32 RBCs may be produced. So from here, kaya mo mag-produce hanggang 8 or up to 32 nucleus. 8 or up to 32 RBCs. Okay? So that's how much red cells you can form from these progenitors, from these cells. So again, the first one that we have here would be your pronormoblast, the earliest recognizable stage. So it's also known as your rubriblast. Again, familiarize yourselves with those terms dun sa table natin. So you have here your pronormoblasts, also known as your rubriblast or your proerythroblast, which is the first morphologically identifiable RBC precursors. Because when it comes to BFUs or CFUs, meron ding CFU, yung mga ibang cells natin. Sila megakaryocytes for platelets, they also have those. So we always start under light microscopy with these recognizable stages. Okay? And as what we have mentioned a while ago, as the cell mature, the size of the cell later on decreases. Tama? So with this pronormoblast, roughly, you can have there about 12 to 20 micrometers in diameter, but this is capable, remember, of mitosis. Okay? It's capable of cell division. And as you can see here on this figure, the cytoplasm is basophilic, but it's dark blue because of the concentration of ribosomes and RNA within your cell. Okay? Yan ang meron tayo sa pronormoblast. And at this stage, remember, you will have here the accumulation of the components necessary for hemoglobin synthesis. As what I've said a while ago, in RBC production, at the end of the day, what's important is for you to synthesize hemoglobin. Because red blood cells, the mature red blood cells, are not capable of synthesizing hemoglobin. So they rely from their precursors. So at this point in time, in pronormoblast, you will have the accumulation of all the things that you need for hemoglobin production. Okay? Iipunin mo na. Sir, does it mean I'm starting to synthesize hemoglobin here? So far, not yet. Okay? Yung entire hemoglobin, ha? So far, not yet. But the globin production, the globin portion, may then begin at this stage. So for you to have an idea, let's have an eye view about hemoglobin. Tandaan, hemoglobin is a molecule made up of heme and globin. Okay? So when they combine, you will form the molecule called as hemoglobin. So sir, why do we produce hemoglobin only among immature cells? Okay? Well, look at this globin first. So sabi natin, globin production may begin on your pronormoblast. So why on your immature cells, sir? On your immature stages? It's simply because these immature stages contain those substances that are needed for hemoglobin production. For example, globin here is a protein. Okay? So for you to produce your proteins, you will need a lot of chemicals, you may need a lot of substances, you may need enzymes here, so to produce that protein. So among your immature cells, you have there your nucleus. Right? As the cell mature, nawawala yung nucleus na yan. That's why your mature RBC doesn't contain any nucleus inside. Right? So, from this nucleus, as what you know for sure, you have DNA. Okay? So, para meron lang tayong konting glimpse, so sana natatandaan nyo pa from your previous courses, yan, especially in biochemistry. So, from DNA, later on, it can be transcribed to become RNA. Diba? Through transcription, you can form your messenger RNA, right? And from this messenger RNA, later on, you can then translate it to become amino acids. So through their bonds, through peptide bonds, let's say, from these amino acids, you can now form your protein. And that protein is what we call as globin. Sir, are my red cells capable of doing that? No. Why? Because your red cells do not have any nucleus. Your red cells do not have DNA inside. Kaya hindi nila kayang gawin yon. Okay? So that's one. So that's why it's very important na if you want to produce enough hemoglobin within your cells, you provide your maturing RBCs with necessary vitamins or necessary minerals for the development of the nucleus and the maturation of your nucleus. Now, for example, you will need their vitamin B12. You will need their folic acid. Yan, for nuclear maturation, for DNA synthesis, you need such vitamins. So if you want to produce this globin to form your hemoglobin, dapat meron tayong normal na supply nun. Okay? And then on the other hand here, heme is made up of two substances known as protoporphyrin 9 and the other one is iron. That's how significant iron is. Without that iron, o parang domino effect, without that iron, you don't form heme. Without heme, you don't form hemoglobin. Without hemoglobin, your red cells will lose its ability to transport and carry your oxygen. So basically, that's the reason on why your hemoglobin is only synthesized among immature cells. Okay? So going back with your pronormoblast, again, the substances needed for hemoglobin production starts to accumulate at this stage, and globin production may then begin at this period. And because of that accumulation of different chemicals needed for hemoglobin production, the next thing you'll have would be your basophilic normoblast or prorubicite. So on this at this stage, your chromatin begins to condense. Ayan, there will be clumps along the periphery of your nuclear membrane, and your chromatin becomes more and more coarse. And you'll have there, ayan, so what you see, somehow a centrally located nucleus. And obviously, you call it as such, basophilic normoblast, because the color of the cytoplasm is intensely blue. Okay? The cytoplasm may be bluer than your pronormoblast. That's why you call it basophilic normoblast. Okay? It's also capable of cell division. It's capable of mitosis. And at this point in time, at this stage, there will then be some sort of detectable hemoglobin synthesis. Sir, if I view it under the microscope, will it be visible? Take note, not yet. You can detect in certain testing presence of hemoglobin or synthesis of hemoglobin, but they are not visible. Okay? This hemoglobin is not visible because of the organelles within your cells, like your ribosomes o kaya yung mga RNA natin. They are not visible under light microscope. But take note, there will be detectable hemoglobin synthesis at this stage. And just like your pronormoblast, it could last for about 24 hours. Yan, si pronormoblast natin lasts slightly more than 24 hours, similar with your basophilic normoblast. Okay? And when you go into the next stage, polychromatophilic normoblast or rubricite rather. Just looking at the term, polychromatophilic. Sir, I now have multiple colors in my cytoplasm. That's why you call it polychromatic or polychromatophilic. So from bluish, there will now be a tinge of gray. And more importantly, there will be a tinge of pink. So meaning, at this point in time, you can now start to see hemoglobin within your cell. Okay? It simply indicates, because of that pinkish colored cytoplasm, it indicates na hemoglobin has already been synthesized. Although hindi pa siya ganun karami, but this time, it's now visible under the microscope. And take note, your polychromatophilic normoblast is the last stage capable of mitosis. The last one that will undergo cell division. Okay? And at this point, parami na ng parami yung sinisynthesize natin na hemoglobin. That's why you can now start, ayan, hopefully you can see, you can now start to see those grayish or up to pinkish colored areas on your cytoplasm. And by the numbers, it may last for approximately about 30 hours within your bone marrow. Okay, so after 30 hours, it will then mature to become orthochromic normoblast or metarubricite. Take note, orthochromic normoblast or metarubricite is the last stage in erythropoiesis to contain the nucleus. Okay? The nucleus is condensed. Yan, may pagka-pycnotic na siya. So, pycnosis will then later on be followed by karyorrhexis. And late in this orthochromic normoblast stage, the nucleus, remember, the nucleus here, that's why it is eccentric. Parang nasa gilid na. The nucleus, remember, is ejected from your cell. the nucleus moves to the cell membrane and into a pseudopod-like projection because you need to let it out. Okay? Lumalabas yan because you then lose vimentin. Yan. Do not forget this. You lose vimentin, which is a protein responsible for holding organelles in their location. So dahil nawawala na yan, nawawala si vimentin natin, your nucleus now can be easily ejected. Okay? The nucleus containing projection separates later on from your cell by having the membrane seal and later on pinch off the projection with the nucleus enveloped by a cell membrane. So paglabas niyan, it is enveloped by a cell membrane. And once out, o kunyari, this is your nucleus, the enveloped nucleus. Once out, it will then be called as the nucleus. I'm referring to the nucleus. Once out, the extruded envelope nucleus is what we call as pyrenocyte. And that pyrenocyte will later on be removed by the macrophages within your bone marrow. Kasi wala naman ang gagawin yan. Pyrenocyte is the extruded nucleus. So because you lose vimentin, these organelles now, yan, your nucleus now, have been ejected. And then later on, that ejected envelope nucleus is then called pyrenocyte. And ang gagawin natin sa pyrenocyte is to phagocytize it. Okay? It will be engulfed by your bone marrow macrophages. So, sir, question. Am I still synthesizing hemoglobin at this stage? Yes. Okay? You are still capable of producing hemoglobin at this stage. Although, at this point, hemoglobin synthesis decreases, but the fact still remains na orthochromic normoblast or metarubricite is capable of producing your cells. Capable of producing hemoglobin, rather. Okay? And the next one here, so after roughly 48 hours, ayan, magstay siya as orthochromic normoblast roughly about 48 hours. So after that, it then matures as the nucleus comes out, o wala ng nucleus, what you will have now will be your reticulocytes. Okay? Your reticulocyte or polychromatic RBCs. Polychromatic erythrocyte. Kasi if it's a mature red cell, remember, if it's a mature red cell, it's just pink. If you look at your smear, it's just pink. But if you have there, let's say, on your blood smear, sir, it's a red cell. But the color is polychromatic. Ang dami niyang kulay, sir. May pagka-bluish, may pagka-grayish, then may pagka-pinkish. Most likely, that cell you are referring to on your smear is a reticulocyte. Okay? Now take note. Reticulocytes are immature cells. Okay? As the rule goes in hematopoiesis, only mature cells will then circulate in your blood. Okay? RBC, yung mga yung mga neutrophils natin, so on and so forth. Only mature cells will then circulate in your blood. All those immature forms will then stay within your bone marrow, except for some. And one of that would be your reticulocytes. Yes, they are immature, but they can normally be present in the blood. And if it's in your blood and you examine your blood smear, it will then appear to be polychromatic as compared to your red cell. If it's red cell, it's just pink. If it's reticulocyte, it's polychromatic. Okay? And as you see here in this picture, your reticulocyte no longer contains the nucleus because it has been ejected out. Right? But take note, what makes it special is that, yes, they don't have the nucleus, but they are still capable of synthesizing hemoglobin. Okay? They can do that. They can still do it. Because inside yan, yung mga nakikita nyo na yan, inside, they have remnants of RNA. Okay? You don't have the nucleus, you just have a little bit of your mitochondria and ribosomes, but more importantly, you have that RNA that can then help you synthesize your hemoglobin. Kaya at this point, we can consider it as the last stage capable of synthesizing hemoglobin. Okay, ang paulit-ulit natin kanina, yung RBC hindi na kaya. Okay? So the last one, capable of synthesizing hemoglobin, will then be your reticulocytes. And it will stay within your bone marrow for about 1 to 2 days. Let's say this is your bone marrow and this is your retics. So once you form retics, yan, you produce hemoglobin. It stays within 1 to 2 days within your bone marrow, and then later on, it moves to your peripheral blood. And after about 1 day, tapos na isang araw, after 1 day of staying in your blood, that's the time it fully matures to become an RBC. To become a fully mature red blood cell. Okay? In the laboratory, let's say you have a whole blood specimen and you want to count reticulocytes. If you want to easily check on these cells and demonstrate these remnants of RNA, you should be using a special stain. You call it as supravital stain. And when you say supravital stain, you are staining the cells while they are living. Okay? Unlike the ordinary staining of blood smears. Yeah, so ordinary staining of blood smears, before you stain it, you fix it first with methanol. So dito, you allow first your blood to come into contact with your stain, and then later on, you prepare the smear. So supravital is staining your specimen while the cells are living. Okay? And as to its importance, reticulocytes, remember, counting them is an important indicator of your bone marrow function. So if you want to know, or if you want to determine the erythropoietic activity of your bone marrow, or let's say if there is an effective erythropoiesis ongoing in your body, you should count reticulocytes. Especially in cases of hemolysis. If you want to know whether this bone marrow is responding or not, you count reticulocytes. Okay, let's have a scenario. Let's have an example. Let's say your patient here has hemolysis. Your patient here has hemolytic anemia. For whatever reason, let's say the red cells are being lysed because of, or let's say, for example, because of parasite, or because of malaria, let's say. So your red cells, the patient's red cell is being destroyed. So usually, what your body will do is to compensate. Parang iisipin ng marrow natin, my red cells were destroyed, so I need to do something. I need to produce more cells. I need to create more RBCs. That's the response of your bone marrow. That's the compensation mechanism. Ang tanong is, sir, how will I know if my marrow responded or not? How will I know if my bone marrow is reacting to this hemolytic process that I have? So the best way to do it is to count for your reticulocytes. Because in cases of, let's say, hemolysis here, in cases of hemolysis, your marrow will create more cells, and what they will do is release these reticulocytes faster than the usual pace. Diba sabi natin kanina, Once you form retics, it will stay in the marrow for about 1 to 2 days before they come out, right? Before they go to your blood. So dito, well, isang araw pa lang, lalabas na si retics. Because your bone marrow tries to respond. Your bone marrow is compensating. Okay? So what will happen now is that as you check your blood, you will then see a lot of reticulocytes. So if this patient here has hemolysis or hemolytic anemia and your marrow is responding, or in short, your marrow is helping you should expect an elevated reticulocyte count. That's a signal na your bone marrow is helping you, is compensating, is responding to your anemia, to your hemolysis. Okay? But then on the other end, let's say you have a hemolytic process, you have hemolytic anemia, let's say, and as you count reticulocytes, it's low. O ibig sabihin nun, there is a problem with your bone marrow functions. There is a problem with the erythropoietic activity of your bone marrow. So laboratory-wise, that's how significant counting reticulocytes is. Okay? It tells you how effective your erythropoietic cycle is. It's an indicator of your bone marrow function. Okay? And then after that, you will then have your RBCs or your erythrocytes. Roughly about 6 to 8 micrometers in diameter and lasts within our peripheral blood for the next roughly 120 days. Okay? Normal RBCs here, normal RBCs here will have that 1/3 central pallor area. The rest is red or pink because of the hemoglobin, and you will have here a 1/3 central pallor area. So that gives your red cells an extra surface area so that they will not be easily lysed. Okay? And once formed, your red cells now will then deliver oxygen to your tissues. They will release it and return to your lungs to later on be reoxygenated. Kasi it's our lungs that will oxygenate your cells. Okay? And on our next discussion, sa RBC, we'll further deal with the structural components of your RBC. So aside from hemoglobin, what are the other things that your red cells need for them to survive and for them to be able to do their job? Okay? So listed on this table here would be the specific characteristics of those cells, of those maturing cells in erythropoiesis. Okay, so hindi ko na babasahin isa-isa ha. So those would be the size characteristic, the NC ratio, and so on and so forth. Okay? Then next one that we have here is your leukopoiesis. Leukopoiesis because it's then the production of your leukocytes. So white blood cells, from the term, you might be wondering, sir, bakit ba tinawag yan na white blood cells? WBCs are called as such because technically, WBCs are colorless. Technically, so relatively, they are colorless as compared to your RBCs. And these WBCs, as what you know, could then be differentiated into granulocytes and mononuclear cells. Granulocytes will then refer to your neutro, to your eosinophil, and to your basophil. Okay? Mononuclear cells will then refer to your monocytes and to your lymphocytes. Okay? So if you will go with the lineages, lymphocytes will be from your lymphoid stem cell, right? And the rest will then be on your myeloid stem cell. And just like with your RBCs, definitely you will need growth factors. You have there your GM-CSF. You have your GM-CSF, your macrophage colony stimulating factor or MCSF. You have your interleukins, 5, 11, interleukin 3, and also KIT ligand. Okay? You will also have that kit ligand. And to promote lymphoid progenitors, to promote lymphoid differentiation, yan, you will have their interleukin 7, o dagdag nyo si interleukin 2, interleukin 12, and interleukin 15. And up to some extent, other interleukins like 13, 14, 16 may also be included. Okay? So for them, for these lymphoid progenitors, it's more of your interleukins, different numbers of interleukins, and on your myeloid, so you will have interleukins together with other growth factors. Yan, particularly sila colony stimulating factor natin. Okay? And the number of the white cells that you will then have, remember, varies from one person to another. As to its differences, it could vary according to the sex, to the age of the patient, to the ethnicity sometimes of the patient. Yan, as to some extent, it could also be within the activity of the patient or within the time of the day when you have collected the blood. Okay? So, to begin with, let's have first your myeloid lineage. So from your myeloid lineage, what you can have here are your granulocytes together with your monocytes. Okay? So the myeloid lineage gives rise to those basophil and monocytes. And as you look at this diagram here, it technically does not show changes in cell and nuclear morphologies. Okay? Technically speaking. So you have here your GM-CSF, ano natin, granulocyte monocyte or granulocyte macrophage colony stimulating factor natin, your GM-CSF, which then stimulates the proliferation and differentiation of your neutrophil and macrophages colonies from your CFUs. Okay? So meron din tayong CFU, hindi lang sa RBCs. So from your pluripotent hematopoietic stem cell, it will then give rise to your forming units, just like your colony forming units, and because of your growth factors like CSF, that's the time you can now start creating and start the development of your cells. Okay? So we have here your neutrophil development. And as to studies, neutrophils share a common progenitor with your monocytes. Okay? And it is distinct from your eosinophils and your basophils. So kaya, ang tawag natin sa progenitor na yan is your granulocyte monocyte progenitor or GMP because they share a common progenitor. And the main stimulant here, the main regulator here for the production of your neutrophils would be your GCSF, your granulocyte colony stimulating factor. And within your bone marrow, yan, look at this figure here, you will have 3 different pools. Okay? You will have here your stem cell pool. Obviously, that's where you have your hematopoietic stem cell that is capable of self-renewal and differentiation. Okay? Then you will have here your proliferation pool or mitotic pool. Obviously, as the name suggests, this would be the stages capable of mitosis. They are capable of cell division. Okay? So you will have here your common myeloid progenitor. Then binanggit natin kanina, you'll have your granulocyte monocyte progenitor. And then, the earliest recognizable stage will then be your myeloblasts. Okay? So from your common myeloid progenitor up to your myelocyte, that will then be your proliferation pool. or mitotic pool. Proliferation because nagpaparami ka na. Okay? And when you reach your metamyelocyte until the neutrophil stage, you then call it as your maturation stage. Okay? Your storage pool. It consists of those cells that undergoes nuclear maturation that form the marrow reserve and are available later on to be released into your peripheral blood. They continuously mature, especially the nucleus, until they are reached into your blood. Okay? Under light microscopy, o parang sa RBCs din kanina, if you look at these things, under light microscopies, they are not recognizable. Meaning to say, they are not distinguishable from one another. O parang sa RBCs a while ago, if you start with this stage, it will then be your earliest recognizable stage. Okay? So the earliest recognizable stage in granulopoiesis, yan, particularly in neutrophil development, is what we call as your myeloblasts. Okay? So you have here a large, look at this figure here. You have here a large, round, and or oval sometimes, round nucleus or oval nucleus which occupies almost the entire cell. Yan, as you see in this picture here. And this myeloblasts here could further be divided into three. You have type 1, type 2, and type 3. Okay? In type 1 myeloblast, remember, it doesn't contain any visible granules if you view it under light microscope. So let's say you have a bone marrow specimen, and then you stain it with Romanowski stains, and you start observing it under that light microscope. In type 1, halos wala ka pa makikitang granules. In type 2 and type 3, that's the time, your cell now will show granules. So those are your granular stages. Okay? In type 2, you will have there your primary azurophilic granules in the cytoplasm. And then in type 3, it could be somehow rare in your normal bone marrow, but they can be seen in certain types of leukemia. Yung type 3 natin. Okay? So the next one here, by the way, they are capable of mitosis. They will divide, and as they divide, in turn, it will then give rise to your promyelocyte. And on this promyelocyte here, look at this one here. There is this paranuclear halo, or we call it as HOF. Paranuclear halo or paranuclear HOF, which is usually observed in promyelocytic stages. Ayan, this one. Ayan. Paranuclear HOF na yan. Okay? The cytoplasm of this cell is quite basophilic. It's evenly basophilic, and it contains, take note, your primary granules. If in red cells, just to compare, if in red cells, among your maturing stage, ang importante is you synthesize hemoglobin. O dito in white cells, ang importante is the appearance of your granules. Okay, kasi those granules will be the one to contain those enzymes needed by your WBCs, by your granulocytes specifically, so that they can then later on digest or kill an ingested foreign body or unwanted material. Okay? So on this promyelocyte, you start to observe for your primary granules. And these are the examples of your primary granules, which are formed during your promyelocyte stage. Okay? So, in other terms, we call this promyelocyte granules, these primary granules also as your non-specific granules. So, you have myeloperoxidases, yung mga elastases natin, lysosomal enzymes, cationic antibacterial proteins. So, these are your non-specific granules. Okay? So on to the next one. So if you have your primary granules there, on to the next one on your myelocyte stage. So this is the time where you can now have your secondary or your specific granules. Okay? Under this myelocyte stage. And take note, myelocyte stage is the last one capable of cell division. It's the last stage in your granulopoietic development that will undergo mitosis. Okay? So at this point, the production of your primary granules later on decreases until it totally stops. And then later on, your cell will start to produce now your secondary or your specific granules. And this secondary neutrophilic granules later on slowly spread through the cell until the cytoplasm is somehow more lavender pink than bluish. O, kasi kanina may pagka, ano pa siya, di ba? Bluish yung color niya. But this time around, as to the appearance of those secondary granules, it's now more of lavender pink than being blue. So listed here are the examples of your secondary specific granules for your neutrophils. Okay? And then on your metamyelocyte stage or your juvenile cell here, the nucleus is then indented, having that some sort of kidney bean shape or peanut shape appearance. And at this point, this is now where you start to synthesize your tertiary granules. So primary, secondary, and then your tertiary granules. Tertiary granules, or also known as your gelatinase granules. Okay? You call it as gelatinase granules. And before it becomes your neutrophils, you will then undergo the stage called as band stage or band cell. Okay? Roughly about 9 to 32% of the nucleated marrow cells and 0 to 5% of the nucleated cells in your peripheral blood will be your band cells. Sir, immature ba yung band cell na yan? Yes. Pero just like with reticulocytes, they can be present in the blood. As mentioned, 0 to 5% in the blood. Marami sa marrow. 9 to 32% of the nucleated cells in the bone marrow will be more of your band cells. Okay? And at this point, the secretory granules, yan, sometimes known as your secretory vesicles, may then begin to be formed at your band cell. Laboratory-wise, take note laboratory-wise, if you are in doubt, kunyari, nag-smear examination ka since band cell is normally present in the blood. If you are in doubt kung ano bang cell yan, so you just call it as segmented neutrophil. Okay? Yan, according to CLSI standards, yan, idagdag lang natin, band cells should be included within your neutrophil count and not reported as a separate category because of the difficulty in the reliability of identifying these cells. So ayan, sabi ng CLSI, if you see a band cell, you can then include it on your neutrophil count. Okay? So these are the secretory granules that we have, or the secretory vesicles that we have, that will later on be used by your neutrophils for their activity. Okay? And then lastly, will then be your neutrophils, your segmented neutrophils. And among all our white cells, neutrophils would be the most abundant. Among white cells, among white cells, this would be the most abundant. It's the highest in your blood. And as what you see here on these figures, you have some sort of rose violet or pinkish colored specific granules. So if you see a cell with nucleus in the blood having that 2 to 5 lobes or 3 to 5 lobes. Yan yung mga lobulations na yan. And you see rose pink colored granules, then you can count it as a neutrophil. And for you to differentiate one WBC from another WBC, specifically granulocytes, what we check basically would be the color of the granules and the appearance of the nucleus. Mostly, ayun lagi yung chine-check natin. Okay? So if you want to know if it's a basophil, an eosinophil, or a neutrophil, check the nucleus and check the granules. Okay? And if you will look at the if you will go with the numbers, myeloblast going to your myelocyte roughly is about 6 days. O lagay natin, myeloblast until it becomes myelocyte, roughly it's about 6 days. Okay? And then later on, the transition time going into your maturation pool. Kasi from myeloblast to myelocyte, that will still be on your proliferation pool, right? Once it enters your maturation pool, doon na sa metamyelocyte, band cell, neutrophil, it will take you roughly about 4 days or up to 6 days more. Okay? And once neutrophil is within your blood, relatively, their half-life will just be about 7 hours. After 7 hours, they leave your blood and they go to your tissues. That's one of the important reasons on why WBC precursors are more abundant in the bone marrow as compared to your RBCs. Yan, you go back with your ME ratio, sa myeloid to erythroid ratio natin. Mas marami tayong myeloid precursors, yan, granulocyte precursors, as compared to your RBCs. Because once in the blood, they will then live within hours as compared to your RBCs that can last up to 120 days. Okay? When it comes to eosinophils, ayan, as written, the development is similar to your neutrophils. The stages that you have there will just be similar. Okay? You will just add a bit of extra interleukins, interleukin 5, yan, interleukin 3, together with your CSF, ha? O kanina, di ba, GCSF lang. So dito, GM-CSF together with interleukins. This time, you can now produce your eosinophils. And as to their development, pareho lang sila halos ni neutrophils. Meron ka ding myeloblast, promyelocyte, myelocyte, metamyelocyte, and after metamyelocyte, eosinophils and basophils, remember, will not undergo the band stage. Band stage will only be applicable for neutrophils. So after metamyelocyte, automatic, it becomes an eosinophil and or a basophil. Okay? And then, on the promyelocytic stage in eosinophils here, pare-pareho lang naman sila ng terms. On the promyelocytic stage, it can be identified cytochemically because of the presence of, sulat ko, kapag eosinophils to ha, because of the presence of Charcot-Leyden crystals on their primary granules. Diba? On your promyelocyte, that is where primary granules appear, right? So to identify them, you can then check for this Charcot-Leyden crystal on the primary granules in your eosinophilic development. Okay? So this is how your eosinophils will then look like. So this is your mature eosinophils. Okay? So again, pareho lang sila ng pareho lang sila ng stages. Then mature eosinophils, yan, as you see on this figure, usually is bilobed, and you will have that reddish-orange colored granules. Okay? So from myelocyte up to your proliferation stage, it will take you about roughly 3 days or 3.5 days bago ka makapag-produce ng eosinophils. And the half-life of eosinophils roughly is about 18 hours within our blood. Okay? 18 hours lang sa loob ng mga blood natin. And then, once in your tissues, these eosinophils can survive roughly about 2 to 5 days pag nasa tissues na natin sila. Okay? So, listed here are examples of the granules you have in your eosinophilic development from the different stages. So, sa promyelocyte, you have your charcot-laden crystal. Sa myelocyte, as you have those secondary granules, this will be your specific granules. Yan, particularly, do not forget this, your major basic protein. Kaya yung eosinophils natin are essential for eliminating parasitic organisms because of these granules, particularly because of that MBP. Okay? And then, lastly, for granulocytes, your basophils will then be somehow similar also to your neutrophilic development. So your basophils are derived from the progenitors within the bone marrow and within your spleen. Then they differentiate under the influence of different cytokines or different interleukins that will then help you produce out your mature basophils. And the population of our basophils could either be IgE dependent or non-IgE dependent. Okay? It depends on the type of mediator response determined by the balance between this population. So if it's an IgE dependent basophil, so more or less, it was under the influence of interleukin 3. If it's a non-IgE dependent basophil, more or less, it's under the influence of TSLP. TSLP stands for thymic stromal lymphopoietin. Again, that is thymic. And for your thymus, thymic stromal lymphopoietin. That's what TSLP stands for. Thymic stromal lymphopoietin. Okay, so that would be for the non-IgE dependent basophils. And basophils, ayan, singit natin, and mast cells are cells with morphologic and functional similarities. Kaya it is believed that once basophils go to your tissues, you then call them as your mast cells. Okay? So you have here the different granules, yan, especially the specific granules among your basophils. Yan, particularly yung histamine natin. Kasi basophils are essential during hypersensitivity and during allergic reactions. Kaya you have those granules. Okay? And then for your monocytes, mabilis lang naman to kasi 3 lang naman yung stages nila. So from your hematopoietic stem cell, ayan, as mentioned a while ago, they share the same progenitor together with your neutrophils. Tama? So you'll have there your common myeloid progenitor and then your granulocyte, macrophage progenitor. Right? So you have that, no? You have the same lang. And once you reach the monoblast stage, that is then your earliest recognizable stage. So from monoblasts, it then matures as a monocyte, promonocyte, and lastly, as a monocyte. And take note that monocytes are the largest cell, blood cell in your blood. Okay? So the main stimulant here, so that your progenitors are committed in producing monocytes, will be your MCSF, your macrophage colony stimulating factor, or monocyte colony stimulating factor. Okay? So again, you have here your monoblasts, you have here your promonocyte. Ang importante dyan is this one, your monocytes. So you have that somehow blue-gray cytoplasm with some sort of ground glass appearance and having that kidney bean-shaped nucleus which could be folded. Sometimes kasi yung nucleus na yan folded, showing brain-like convolutions. And take note, sa mga monocytes natin, you will see these vacuoles. Okay? You will see those vacuoles. And these monocytes may then last within, or may then be produced within 60 hours. 16 monocytes in 60 hours. Okay? So these are some of the locations once monocytes leave your blood. Tandaan, monocytes are just the same as your macrophages. O macrophage if it's within your tissue. Monocyte if it's within your blood. So if it's in the tissues, or say for example, it's in your liver, you then call it as Kupffer cells. If it's in your skin, you call them as your Langerhans cells and so on and so forth. But they function the same. Okay? Si monocyte / macrophage. Okay? Then lastly, just a glimpse lang for your lymphoid. You'll further elaborate this in your immunology class. So you can have 3 major groups on your lymphoid lineage. So you can have here your T cells, your B cells, and your NK cells or your natural killer cells. And to further divide these lymphocytes that you will produce in this lymphoid lineage, it could be dependent on the type of immunity that you have. For T cells, you can have there your cell-mediated immunity. And then for your B cells, that's the time, you can now have your humoral immunity. Okay? Tandaan, B cells and T cells are non-phagocytic. If you will observe them under the microscope, B cells and T cells will just look the same. Okay? Morphologically, they're just the same. So if you will report it on your CBC result, for example, you just simply write it as lymphocyte. Okay? Lymphocyte lang. You don't write it as a B cell or a T cell. Just simply write it as lymphocyte. Morphologically, they are the same. But functionally, they are different. Cell-mediated immunity, you have your T cells. You have there your T helper cells or your cytotoxic T cells. Then, for your B cells, yan. Yan yung for humoral immunity. Kasi later on, that B cell will turn into a plasma cell that will help you produce your antibodies. Okay? So for your lymphocyte development, so pwedeng antigen independent or antigen dependent. So when you say antigen independent, so the development of lymphocyte occurs within the bone marrow and in your thymus. Meaning to say, even without a stimulation, you can then produce your B cells and all your T cells. O kaya, yung bone marrow natin, tsaka yung natin, thymus natin, are known as your, connected into your immune nucleus, but they are known as your primary lymphoid organs. Right? Then when you say antigen dependent from the term itself, lymphocyte development occurs in other parts of your body like your spleen, lymph nodes, tonsils, and your malt. Yan. As to your secondary lymphoid organs naman. Okay? Or because of certain antigen requirement. So kapag sa primary lymphoid organs natin like thymus and bone marrow, it's antigen independent, and because of the antigen stimulation, it could then be on your secondary lymphatic organs. So that is now your antigen dependent one. Okay? So for your B cells here, it will then start from pro-B, pre-B cell, and then your immature B cells. Pag sinabi nating immature B cells, remember, it's not yet exposed. So, meaning to say, it is antigen naive. Okay, so baga wala pa siyang kamuwang-muwang. Nakuha? Later on, because of antigen stimulation, di ba kaya nga kanina antigen dependent, they mature in your secondary lymphoid organs. O kapag walang kahit anong antigen stimulation, that's within your primary lymphoid organ. So that is antigen independent. Di ba? Pag nag-mature na sila with regards to your antigen stimulation, that is then your antigen dependent one, which then occurs on your secondary lymphoid organs. Okay? Then for your T cells, yan, pareho lang naman sila. You have your pro-T, pre-T, and immature T cells. So during these phases, they undergo antigen receptor gene rearrangement so you can produce T cell receptors. Ayan, those T cell receptors are unique to each of your T cells. Okay? O, pag sinabi natin uling antigen naive T cells, those are the one that's within your thymus, produced by your thymus. And later on, they migrate to your secondary lymphoid organs. Okay? Then, lastly, yung isa is yung NK cells natin. Mature NK cell is, yan, as you see on this figure, technically large with other resting lymphocytes because of an increased amount of your cytoplasm. Yan, medyo malaki sila. And the cytoplasm may then contain azurophilic granules which are negative for peroxidases. Okay? Unlike with your granulocytes a while ago. May mga peroxidases sila. But on this one, wala. They have azurophilic granules but negative for peroxidases. Okay? Then, later on, you can now have your lymphocytes. And lymphocytes could either be lymphocytes could then either be small lymphocyte, roughly around 8 to 10 micrometers in diameter, medium lymphocyte, mga 10 to 12 micrometers in diameter, and large lymphocytes, which could be 12 to 16 micrometers in diameter. Okay, so yun yung mga pwedeng possible na lymphocytes na meron tayo. Okay? Then if you go this figure here, yan. Ito na yung mga sagot dyan. So if you identify them, so you will have here your neutrophils having the 2 to 5 lobes or 3 to 5 lobulations with rose pink granules. You have here your lymphocyte. Lymphocyte usually almost has the same size as with your red blood cell. Halos magkasing laki lang sila. And to differentiate it from other white cell, lymphocyte has a very large nucleus that almost occupied the entire cell. Diba? Konti na lang yung cytoplasm niya. And it's, the size usually, diba? You have small, medium, large, but usually it's more on that size comparable almost to your RBCs. Then eosinophils here, bilobe nucleus, although it's not that visible here, but having that rose pink granules. You have here your monocyte with kidney bean-shaped nucleus. You have another neutrophil, another monocyte here. A basophil here, most of the time, is confused as a lymphocyte. Pero remember, if it's a basophil, you will have their darkly stained purplish granules. Okay? May mga granule as compared to your lymphocyte where you can still see a little portion of your cytoplasm. With basophil, you cannot clearly see the nucleus, the lobulation, because it is covered by the darkly stained purplish granules. Okay? And then lastly, for this lineage-specific hematopoiesis is megakaryopoiesis. When you say megakaryopoiesis, it is the production of megakaryocytes. Sir, nasaan yung platelets? Remember, platelets are part of the cytoplasm of megakaryocytes. Platelets are cytoplasmic fragments of your megakaryocytes. Okay? So in short, if you don't produce out these megakaryocytes, you will not produce your platelets. Okay? So a little portion of megakaryocytes may be seen or may be found in your lungs, but majority, remember, will definitely be on your bone marrow. And to stimulate your stem cells, your myeloid progenitor stem cells, to be committed in producing megakaryocytes, definitely you will need growth factors, particularly a growth factor called as thrombopoietin. So kung sa red cell, meron tayong erythropoietin. Dito, meron tayong thrombopoietin. Okay? We'll further elaborate this when we go into hemostasis in your HEMA2 class. But listed here are the progenitors in your megakaryocytic production. The first two, remember, will be needed for your proliferation. Kasi BFU-MEG and CFU-MEG will then be capable of undergoing mitosis or cell division. So from one mother cell, it gives rise to two identical daughter cells. Okay? But when you reach this light density colony-forming unit megakaryocyte, it loses its ability to undergo mitosis. Pero, it retains DNA replication and cytoplasmic maturation. In mitosis, this is your mother cell and you will have two identical daughter cells, right? When you reach this stage, you will undergo a specialized form of division called as endomitosis. And when you say endomitosis, only the nucleus divides. The cytoplasm matures, but only the nucleus will divide. Meaning to say, if this is my first cell, I have a single nucleus. After I undergo endomitosis, it will still be a single cell, pero this time around, multiple nuclei inside. And that's the reason on why megakaryocytes are considered as the largest cell in your bone marrow. Because as they mature, they become larger and larger. Unlike sa RBC kanina, di ba? O kaya sa WBC. As they mature, they become smaller and smaller. This time around, in megakaryopoiesis, because of endomitosis, the cell becomes bigger and bigger as they mature. So parang ano yan? Pag sinabi natin endomitosis, it's a form of cell division which lacks, hopefully you can still remember the term, which lacks telophase. Diba? Sa cell division natin, the stages. It lacks telophase and cytokinesis. So what divides only would be your nucleus. So as they mature later on, as the cell mature, the size increases, the NC ratio decreases, the number of nucleus increases, which will then be essential so that later on, platelets can be shed out. Platelets can come out. As more nucleus you have, it can go as high as 32. As more nucleus you have, the more chances of platelets being shed out from the cytoplasm. Kasi yung platelets na yan, they're just inside. So for you to be able to release them, to shed out, you will need to continuously mature and undergo endomitosis. And from the first stage, you call it MK1 or megakaryoblast, up to your MK3 or megakaryocyte, it will take you about 7 days. O megakaryoblast, MK1, MK2 is pro-megakaryocyte. MK3 then is megakaryocyte. So from that stage, megakaryoblast to the megakaryocytic stage, roughly it's about 7 days. And for every, by the numbers, for every 1,000 bone marrow cells, you'll only have around 1 to 4 megakaryocytes. Ganun lang kakonti. And for every megakaryocyte, it can then give you roughly 2,000 to 4,000 platelets. And of all the produced platelets, remember, for example, gumawa ka ng 1,000 platelet, for example, only 70% circulates. The other 30% will then be sequestered in your spleen. And as to studies, there's no clear reason for that, but as to studies, to theories, later on, when the need arises, your spleen might possibly release out your platelets. But what we know is 70% circulates in the blood, the other 30% will then be stored in your spleen. Okay? And once you reach this megakaryocyte stage, dami mo ng nucleus, that's the time you can then undergo thrombocytopoiesis. Okay? O that means platelet shedding. That's when platelets start to come out from your cytoplasm. And this is how your platelets will look like under the microscope. Platelets will then be the smallest cell in your blood which contains granules that are needed or essential in the formation of your clot. Platelets roughly are just about 2 to 4 micrometers in diameter with 8 to 10 femtoliters in volume. As what you see on the picture, it doesn't contain any nucleus. Meron niyang mitochondria pero wala yang nucleus. In vivo, remember, in vivo, without any injury, platelets are round. Because as platelets circulates in our body, remember, they are inactive. We only activate platelets if there is an injury. We only activate them when there is bleeding. And when platelets are active from discoid shape, pag inactive sila, discoid sila. From round-shaped platelet, if you activate them, they could then change their shape and assume irregular appearance. Pag walang injury, it's something biconvex in vivo. May pagka-roundish. Pag merong injury, they will change their shape so they can help you form the clots. Okay? So to sum it up, in this lineage-specific hematopoiesis, remember, important thing here is we allow our stem cells to be committed in producing specific cells. Okay? And we do that by providing them stimulants or growth factors that will allow them to produce a specific cell. Kasi from your stem cell, kaya nilang gawin lahat yan. Okay? But at the end of the day, they need to be committed with only one. They need to be specific with only one. That's why hematopoiesis always is regulated and controlled. Do not forget that. Your body will produce what you need. So that's why these stimulants, these growth factors, you also have their inhibitors that will make sure that hematopoietic cycle is always on the right track. Your body should produce what you need. If there is a need to increase your WBCs, then your body will do it. In response, for example, to infection. If there is a need for you to release more RBCs, your body will do it in response to bleeding or in response to hypoxia. So hematopoiesis, remember, should always be controlled and regulated. Okay?